Welcome to the second part of gram-positive bacilli. In this part, we'll be going through the genus Bacillus. Bacillus is a gram-positive bacilli, but it tends to decolorize very easily. So if we are dealing with older cultures, we can have gram-variable bacilli in gram staining, or even sometimes frankly gram-negative bacilli also. The genus Bacillus produces non-bulging spores, and these spores are ubiquitous, and they are found in soil, dust, air, water, and are the commonest contaminants of bacteriological cultured media. Most of the species of bacilli are motile with peritracheous flagella except Bacillus anthracis. The medically important species of Bacillus are Bacillus anthracis and Bacilluscereus. Coming to Bacillus anthracis, now Bacillus anthracis is exclusively associated with certain features like Bacillus anthracis was the first pathogenic bacterium to be observed under the microscope. It produces the disease called as anthrax, and anthrax was the first communicable disease shown to be transmitted by infected blood inoculation. Then Bacillus anthracis was the first bacterium to be isolated in pure culture and shown to possess spores. It was the first bacterium to be used for preparation of live attenuated vaccines, and it is one of the largest pathogenic bacteria. Come to the morphology of Bacillus anthracis. It is a gram-positive bacillus, but it shows a special appearance which is called as the bamboo stick appearance. In this characteristic appearance, the bacilli are arranged in long chains, end to end, each attached to each other, and each bacilli, the ends of each bacilli are concave and swollen, and the entire chain is covered, or it possesses a capsule. The entire chain is surrounded by a capsule. So this gives us a typical appearance of a bamboo stick. Bacillus anthracis possesses a capsule which is polypeptide in nature, and this capsule is encoded by a plasmid. Capsule is formed in media containing added bicarbonates or 10 to 20% of carbon dioxide. Instead of carbon dioxide, we can also add certain ingredients like sedum, albumin, charcoal, or starch to the medium and enhance capsule or encourage capsule formation. Then Bacillus anthracis has spores. It produces spores. These spores are central and oval in shape. Position of the spore is central and the shape of the spore is oval. These spores are non-bulging, and these spores can be observed by performing special staining techniques like the ash-base methods, in which the malachite green is used, and we can also use the Zn staining method because these spores are acid-fast. And in the Zn staining method, we can use 0.25% of sulfuric acid as a decolorizer. Then Bacillus anthracis possesses fat globules. These fat globules can also be seen by staining them with Sudan black B. This method is called as the Virulence method. Then Bacillus anthracis exhibits the McFadden's reaction. Here, when the blood films of anthrax bacilli are stained with Ger's polychrome methylene blue for 30 seconds, the bacillus takes up a blue color, and the capsular material around it takes up a purplish, amorphous purplish color. This reaction is used for presumptive identification of anthrax in animals. Coming to the cultural characteristics, on nutrient agar, the colonies which appear are irregularly round and dull. These are pretty large colonies of about 2 to 3 mm in diameter, and they are grayish white in color. It gives a typical frosted glass appearance. If the colonies, the edge of the colony is observed under the low power of the microscope, it is observed that the long chains of the bacilli give the appearance of entangled matted hair, okay, which is typically called as the Medusa head appearance. We can also grow them on gelatin stab agar. Here we take gelatin stabs and we stab inoculate the medium. This gelatin stab starts liquefying. The liquefaction is slow. So liquefaction commences on the top, and as we move on towards the bottom, the liquefaction goes decreasing. Okay, so it gives a typically inverted fir tree appearance. Bacillus anthracis can also grow on blood agar producing non-hemolytic colonies. It can also grow on a selective medium called as plet agar. Plet agar consist of polymyxine, lysozyme, EDTA, and thallus acetate. So we have pictures of growth of Bacillus anthracis on blood agar and gelatin stab cultures. Coming to the virulence factor, Bacillus anthracis has two virulence factors. It produces a polypeptide capsule and it has an anthrax toxin. This capsular polypeptide inhibits complement-mediated phagocytosis. It is a plasmid encoded capsule is plasmid encoded. So as long as the plasmid is present in the strain remains. The strain produces the capsule. And on this basis, a spore vaccine is produced, which is an attenuated spore vaccine. This strain is called as the Sterney strain. Next, we have the anthrax toxin. This toxin is also plasmid encoded. This toxin has three fragments, the edema factor or OF or factor 1, which causes edema. Next, it has the protective antigen factor, the PA or factor 2. Third, it has the lethal factor or LF or factor 3. This brings about cell death. Individually, these three fragments are inactive. They become active only when these three components or these three fragments work collectively. Coming to the disease anthrax. Anthrax can be present in animals as well as in humans. Basically, anthrax is a zoonosis. It is a zoonotic disease. The sheep and cattle are commonly affected. These are grazing animals, so these graze on the vegetation around. The spores of the bacilli are present on the vegetation and these spores are ingested by the animals. In this way, the bacilli or the spores enter into the animals and the animals suffer from the disease. They may manifest in the animals, anthrax may manifest as a fatal septicemia. The infected animals shed the bacilli through the discharges from the mouth or nose or rectum. Hence, they remain as a source of infection in the environment. Humans acquire the disease through inoculation of spores or ingestion of the bacilli. So in humans, anthrax can be cutaneous anthrax, pulmonary anthrax, or intestinal anthrax. Coming to cutaneous anthrax, this is also called as the Hyde-Porter's disease. The spores are the spores abrade the spores enter into the human through the abraded skin. Usually the exposed surfaces like the hand, neck, face and arms and the back are affected. The lesion starts with a papule which turns into a vesicle. Later on the whole area becomes congested and it becomes edematous and it turns into a lesion called as a black E-scar. This is a lesion with central necrosis. This black E-scar is also called as a malignant pustule. Cutaneous anthrax resolves spontaneously, but in about 10 to 20% of untreated cases, a fatal septicemia may develop. So this is a picture of the black E-scar. Next, coming to pulmonary anthrax, this is also called as the Wool-Soter's disease. This is due to inhalation of the dust. The dust basically contains the spores of the bacilli. This is a hemorrhagic pneumonia with high mortality, and it may also complicate into hemorrhagic meningitis. Coming to intestinal anthrax, this is acquired by ingestion of animal carcasses which contain the bacilli. Okay, so the animal basically dies because of anthrax and this animal is consumed by humans. This results into a violent enteritis with bloody diarrhea with high mortality. Coming to the laboratory diagnosis, the appropriate specimen should be collected for cutaneous anthrax A swab from the lesion may be collected for pulmonary anthrax, sputum may be collected for intestinal anthrax, feces is collected, and for septicemia, blood is collected. The specimens can further processed for microscopy. Different staining methods can be used. We can use the gram staining method to demonstrate the bamboo stick appearance, or simply to see gram-positive bacilli arranged in chains. We can use the McFadden's reaction to see the amorphous capsular material. We can also use the direct immunofluorescence staining method, which will detect the capsular or the cell wall antigen. Then we can do the culture of the specimen. Culture can be done on nutrient agar, it can be done on blood agar, it can be done on the gelatin stab as well as the plate agar. On each of the media, we can have the characteristic appearances as we have discussed earlier. Then we can also do serology to identify or to detect the antibodies, and we can do the molecular diagnostic tests by PCR to detect the specific gene of the bacilli. Coming to the prophylaxis and treatment, for prophylaxis vaccine is available. Vaccine is available for animals as well as for humans. The animal vaccine is called as the Stern's vaccine. This is a spore vaccine as we have discussed earlier. One dose of vaccine is given to the animals, which gives a protection for about 1 year. In humans, we have an alum precipitated toxoid vaccine, and this vaccine is indicated in the occupationally exposed people. For treatment, antibiotics like doxycycline, clindamycin, ciprofloxacin, and rifampicin work well. Monoclonal antibodies are also available, which can be used in cases of anthrax. Next, coming to Bacillus cereus. This is a species which causes food poisoning. Bacillus cereus is widely distributed in nature, and it can be readily isolated from soil, vegetables, milk, cereals, spices, meat, and poultry. Bacillus cereus is also a gram-positive bacilli. It is motile, but it is non-capsulated. Bacillus cereus grows on a medium called as mannitol, egg yolk, phenol red, polymyxine agar. Bacillus cereus produces the diarrheal type or the emetic type of food poisoning. In the diarrheal type of food poisoning, the person suffers with diarrhea. Vomiting is rare, and in the emetic type of food poisoning, this person suffers with vomiting. Diarrhea is rare. Diarrheal type of food poisoning results after ingestion of food articles like meat, vegetables, and cereals, whereas emetic type of food poisoning results after ingestion of rice, especially the Chinese fried rice is incriminated. Antibiotics like clindamycin, tetracycline work well. With this, we end with a brief discussion of medically important species of Bacillus.